Look at your hand. It is made of cells, billions of them. Inside each cell is a nucleus. And inside that nucleus is a code, a set of instructions. This is DNA. It is the blueprint for you. This elegant molecule, a twisted ladder we call a double helix. It carries the information that built you from a single cell into the complex, thinking being you are today. It determined the color of your eyes, the texture of your hair, your predisposition to certain traits. This is the realm of human genetics, the study of this code, the exploration of the instruction manual for humanity. It is a story written in a language of 4 letters, a TCG, a story that stretches back billions of years, connecting us to the very first life on Earth. Imagine a library containing thousands of books, all written in a language you couldn't read. This was the human genome for most of our history. We knew it was there, but the text was a mystery. Then, in 2003, a monumental effort was completed. The Human Genome Project. For the first time, we had read the entire book of human life from cover to cover. This was a game changer. It gave us a reference map, a guide to our own genetic landscape. Suddenly, we could begin to pinpoint the specific spelling errors, the genetic mutations, responsible for diseases like cystic fibrosis, Huntington's disease, sickle cell anemia. The library was finally open. With this map in hand, scientists developed incredible new tools to navigate it. One of the most powerful is genetic testing. With a simple saliva sample, companies can now analyze parts of your DNA. They can tell you about your ancestry, tracing your family's journey across continents. More importantly, they can identify genetic markers that might increase your risk for certain conditions, from heart disease, Alzheimer's. This is knowledge as power. The power to make informed decisions about your health, to take preventative measures, and to understand the biological hand you've been dealt. It's like getting a personalized weather forecast for your body's future. But what if you could do more than just read the code? What if you could edit it? Enter CRISPR-Cas9. Think of it as a biological find and replace function for DNA. It's a technology adapted from a defense mechanism found in bacteria. CRISPR allows scientists to find a specific segment of DNA with incredible precision, and then cut it, allowing them to either disable a faulty gene or even insert a new correct one. This isn't science fiction, it's happening in labs right now. This tool has revolutionized biological research and opens the door to directly correcting the genetic errors that cause devastating diseases. These breakthroughs, genome sequencing, genetic testing, gene editing, represent a quantum leap in our ability to interact with our own biology. They are the new toolkit of the 21st century. They have transformed human genetics from a field of observation into a field of intervention. We are no longer just spectators of our genetic fate. We are becoming active participants with the ability to read, diagnose, and even rewrite the very code of life. This new power is exhilarating, but it also places a heavy burden of choice upon our shoulders, a burden we must now learn to carry. The most powerful argument for advancing human genetics is the immense potential for good. It's the promise of a world with less suffering. Think of a child born with a severe genetic disorder, like Tay-Sachs disease, where a single faulty gene leads to a progressive and fatal deterioration of the nervous system. For centuries, medicine could do little but offer comfort. Now gene editing technologies like CRISPR offer a glimmer of hope. The possibility of correcting that single genetic error before it can wreak its havoc. We could potentially turn a death sentence into a manageable condition, or even a complete cure. This isn't just about extending life, it's about improving its quality in the most profound way. Consider the diseases that haunt our later years: cancer, Alzheimer's, heart disease. Many of these have strong genetic components. By understanding the genetic risk factors, we can develop more personalized medicine. Instead of a one-size-fits-all approach, treatments could be tailored to your specific genetic makeup. With great knowledge comes great vulnerability. As we unlock the secrets of our genome, we create a new category of personal information, one that is more intimate and revealing than any other. And with it comes the risk of a new kind of prejudice: genetic discrimination. Imagine applying for a job. You are qualified, you are experienced, you are eager. But your genetic test, taken for health reasons, reveals a predisposition to Parkinson's disease later in life. What if an employer saw that information and decided not to hire you, fearing you might become a liability in 20 years? You're healthy now, but judged on a future possibility written in your DNA. This isn't just hypothetical. Consider the world of insurance. Life insurers assess risk. With genetic access, they could calculate your odds of developing cancer. They could calculate your odds of developing heart disease. and they do it with alarming accuracy. They might charge much higher premiums, or even deny you coverage altogether, based on genes you were born with. This creates a system where biological disadvantage becomes financial disadvantage. Your genetic makeup, something you can't control, could block securing your family's future. Laws like the Genetic Information Non-Discrimination Act, GINA, in the US, offer some protection. You spit into a tube and mail it off. A few weeks later you get a fascinating report about your ancestry, about your health risks. It feels personal, a journey into yourself. But what happens to your DNA sample and the data extracted from it? Where does it go? Who has access to it? This is the central privacy question of the genetic age. Most direct-to-consumer genetic testing companies have privacy policies. Those policies can be long and complex, and they're often subject to change. By agreeing to terms, you may grant the company the right to de-identify your data, use it for research, or even sell it to third parties, like pharmaceutical companies. Your genetic data is a treasure trove for researchers. It helps find new drug targets and understand diseases at scale. But de-identified isn't always anonymous. Scientists have re-identified people from anonymized genetic databases by cross-referencing public info on genealogical websites. So far we've talked mostly about using genetic technology for healing, to fix what's broken. But the same tools that can correct a gene for a deadly disease could also be used to alter other traits. Designer babies. What if parents could choose their child's eye color, or make them taller, or enhance their intelligence, or enhance their athletic ability? This is the controversial idea of designer babies. It moves us from the realm of therapy to the realm of enhancement. It's the difference between ensuring a child is born healthy and ensuring they are born with specific desirable characteristics. This is where the ethical landscape becomes incredibly complex and emotionally charged. Proponents of genetic enhancement argue from a position of parental liberty and procreative beneficence. They might say, if we have the power to give our children the best possible start in life, why shouldn't we use it? Parents already try to do this through education, nutrition, and providing a safe environment. Genetic enhancement, in this view, is just another tool to help a child flourish. They might argue that enhancing traits like intelligence or resistance to disease is a good thing, not just for the individual but for society as a whole. A smarter, healthier population would be better equipped to solve the world's problems. There are two fundamental ways to edit human genes. The first is somatic editing. This targets the body's cells, lung cells, blood cells, to treat a disease in a single individual. These changes are not heritable. They die with the person. The second type is far more consequential, germline editing. This involves altering the DNA of a sperm, an egg, and embryo. These changes become part of the resulting person's entire body, including their own reproductive cells. This means the edit will be passed down to all of their descendants. It is a permanent alteration to the human gene pool. The argument for germline editing is one of ultimate prevention. Why treat a hereditary disease in every generation when you could eliminate it from a family line forever? If you could edit an embryo to remove the gene for Huntington's disease, you wouldn't just be saving one person. We find ourselves at a crossroads in the human story. In one direction lies a future where we harness the power of genetics to conquer disease, alleviate suffering, unlock new potentials. In another, a future of deeper divisions, unforeseen consequences, loss of what we consider fundamentally human. The tools are on the table. The code is in our hands. The choice of which path to take is ours. We have explored the incredible promise of this technology, the hope of a healthier world, free from the tyranny of inherited illness. This is a powerful and noble goal that drives the engine of discovery. But we have also peered into the shadows: genetic discrimination, privacy paradox, ethical dilemmas of enhancement, designing our children, permanently altering the human gene pool. These are not simple problems with easy answers. They are deep, complex challenges that force us to question our values, our sense of justice, our vision for the future of our species. What choice will we make?